Hello and welcome back to another MCAT ACE lecture. Today's lecture is going to focus on the immune system. We are more specifically going to focus on the difference between innate and adaptive immunity and then discuss different components of these modes of immunity. Discuss the definition of clonal selection, discuss the structure of antibodies, and then explain how by picking up different antigens from surrounding environments, the body learns to make antibodies that are specific for that antigen. Then discuss the autoimmune disorders, what's the reason that these autoimmune disorders are being developed, and then finally discuss different immune tissues and cells. So, starting with different modes of immunity. So basically the immune system consists of different organs and different cell types that protects our bodies against microbial infections. And these line of defenses are divided into two main divisions. The first one is innate immunity, which is basically present from birth and is neither learned nor adopted. And then the second mode of immunity is adaptive immunity, which becomes activated once antigens from the surrounding pathogens are being captured and processed by our immune cells. So let's go back to innate immunity and discuss different components of this type of immunity. So innate immunity is the first line of defense that is present from birth and is neither learned nor adopted. And these defenses could include not only cells but also resistant barriers. As an example, skin is a resistant barrier that protects against invasion by pathogens, and that's why when your skin is cut, now there is increased risk of infection. There are also oil and sweat glands that maintain an acidic skin environment to inhibit growth of microorganisms. And then finally, our sweat glands are secreting the enzyme called lysozyme, which digests bacterial cell wall. The next mode of innate immunity are macrophages, which ingest microbes by phagocytosis. So macrophages will ingest microbes by phagocytosis. And so once this bacteria is being enclosed by a membrane-bound vacuole and enters the cell, that's the time that lysosome enzymes will be released to digest and kill the microbe. So after phagocytosis, lysosome enzymes will be released to kill and digest the microbe. Now I should mention that in addition to microbes, macrophages are also capable of engulfing, like for instance, viruses, cellular debris, and other dust particles. So macrophages are not necessarily specific to microbes. The next mode of innate immunity is induced by neutrophils, and these are very similar to macrophages in that they engulf and kill their bacterial cells, but they also release free radicals that will kill the surrounding bacteria. So they will both phagocytose and also release free radicals. And neutrophils are the component of pus. So once you have an infection and you say that there is pus formation, it's all the dead neutrophils that you find in that solution that is referred to pus. The next mode of innate immunity is induced by natural killer cells, and these are specific to cancer and viral infected cells. So our cells, as I will discuss later on, they have specific antigens at their surface that will be recognized by our immune cells, and consequently the immune cell will not attack them. Cancer and viral infected cells, however, will have different receptors, as a consequence of which natural killer cells will now know that these are abnormal. They are not our normal cells, and so will target cancer and viral infected cells. And the way that actually natural killer cells will destroy these infected cells is that they would release perforin, which will digest into the cell membrane and form a pore, as a consequence of which now water will flow into the pore of these cells and cause swelling and burst of these abnormal cells. The next component of the innate immunity is complement factors, which are basically molecules that are freely circulating in the blood, and once these complement molecules come in contact with a pathogen, they would aggregate to form a membrane attack complex, which is now capable to insert itself into the membrane and cause formation of membrane pores. So complements form membrane attack complex, as a consequence of which it will form pores within the membrane. The next mode of innate immunity is by interferons, which will inhibit viral replication and protein assembly. And then finally, the last mode of innate immunity is C-reactive protein, which stimulates different immune pathways. And this is in fact an inflammatory molecule that is now able to stimulate different immune pathways. Now, as we just discussed, this innate immunity is basically the initial response to microbial invasion, but it's not very specific. It's just a general mode of attacks. And so therefore, some microbes can evolve strategies to overcome these innate immunities. Under these circumstances, we need a more potent and more special immune attack, which is now referred to as adaptive immunity. And actually, innate immunity does have a role in initiating adaptive immunity, and it's by the fact that macrophages and dendritic cells, these are the immune cells that will pick up the antigen and now present them to T cells, as a consequence of which now adaptive immunity can get activated. So now let's go over adaptive immunity, the other name for which is acquired immunity. So adaptive or acquired immunity. So this type of immunity, as we just discussed, will become active when an antigen is being picked up by antigen presenting cells, like for instance by macrophages or by dendritic cells, and then they are being processed and presented to T cells. So antigen is being picked up by antigen presenting cells, examples of which include, for instance, macrophages or dendritic cells, and now these antigens are and now via these cells, these antigens are being presented to T lymphocytes to initiate an adaptive immunity. So after this point, once T cells are activated, they could mount two different forms of immunity. The first form is called a cellular immunity, which is mediated by CD8 positive killer or cytotoxic, that's the other name for them, T cells. And then the other mode of immunity is humoral immunity, which is mediated by B cells, so B lymphocytes or plasma cells. And this type of immunity will release antibodies. So the cellular immunity activated killer T cells that will now destroy the pathogens, versus humoral immunity initiates release of antibodies. And so in order for you to better understand how the mechanisms of cellular versus humoral immunity is working, we will first have to discuss the function of the major histocompatibility complexes, which are basically the type of glycoproteins that are present on the surface of our cells. And these genes have many different forms, and so every person has their own unique MHC molecules, which make it possible for our immune system to know, okay, this shape of MHC belongs to my body, I will not attack this MHC, but any other glycoprotein forms, or in other words, any other major histocompatibility complex, that I do not recognize, I will attack. So there are two types of MHC glycoprotein. The first one is MHC1, which is present on all of our immune cells, versus MHC2 is only present on antigen presenting cells. So with the MHC1, our immune cells recognizes them and doesn't mount an immune attack. But if our cells are being infected by either cancer cells or by viruses, what happens is that now this MHC molecule changes and it's not going to be the same as the normal cells, as a consequence of which the cytotoxic cells will recognize this abnormal MHC1 and mount an immune attack. So here I've shown you that this type of MHC1 is being used for elimination of infected or cancer cells that are being recognized by CD8 positive T cells, or CD8 positive, which is the same as cytotoxic killer cells. The reason we say that the peptide bonding here is endogenous is that the protein that will bind to MHC1 and is expressed on the cell surface, it has been produced by the cell itself. It's not like the cell has picked up an antigen. So inside the cell is corrupt and infected. And what is being presented on the MHC1 is a warning sign to tell our CD8 positive T cells, the other name for which is killer or cytotoxic T cells, to attack this cell and kill it. And unlike MHC2 that can bind invariant chain, and I will explain later on what it is, MHC1 molecule doesn't bind the invariant chain. So this was an example of MHC1, how it helps with the recognition of infected cells by cytotoxic T cells. Okay. Now the next major histocompatibility complex is class 2, that will help the cells to recognize antigens and mount an attack. So this type of antigen is only present on antigen presenting cells, like for instance macrophages, dendritic cell, and B lymphocytes, and it's being presented to helper T cells or CD4 positive T cells in order to mount an immune reaction. So it's being recognized by CD4 positive. The reason we say that the peptides that are bound are exogenous is that unlike the cancer infected cell that the protein from inside was being processed on the surface, here with MHC2 we are actually processing an antigen that was picked up and then it has entered the lysosome. Now I just told you that MHC class 2 has an invariant chain, and basically what this invariant chain does is that it prevents binding of self proteins or self antigens to it. However, once the MHC2 enters the acidic environment of endosome, that's when the invariant protein can come off and any antigen that is in that environment can bind to it. And then later on, MHC2 will go to the surface and get recognized by CD4 positive helper T cells. Now, once CD4 positive helper T cells become active, they can now either activate more CD8 positive T cells, or they can activate B cells to produce antibodies against this type of antigen that they have recognized. So one memory aid that I have here for you to remember that MHC1 is being recognized by CD8 positive T cells is that 1, so MHC1 times 8, which is CD8 positive cells, is equal to, and then you recall I told you MHC2 is being recognized by CD4 positive T cells, so equals 2 times 4. So all these values are equal to 8. And what this means is that MHC1 is being recognized by CD8 positive cytotoxic T cells, while MHC2 is being recognized by CD4 positive helper T cells. And here I have shown you the memory aid, rule of 8. Now, the next question is, the CD8 positive T cells attack the cells that have expressed abnormal MHC1 because they knew that these cells are somehow corrupted or infected. Now, what happens after CD4 T cell activation? So now the T cell has become activated and it can turn into long-lived memory T cells, so that in future if the same antigen is being recognized, this T cell is now ready. It's like, I know what you are, I know how to defend against you. So this T cell will activate CD8 positive killer cells or killer T cells, and it will also activate B lymphocytes, which will turn into plasma cells, and plasma cells will produce antibody. So here I have provided you with a summary that shows you what happens after antigen is being picked up by an antigen presenting cells, like for instance macrophages. These exogenous antigens are being presented on MHC2 receptor, which is being recognized by the CD4 positive T cells. So remember, 2 times 4 equals 8. That's the rule of 8. Now CD4 positive T cell will activate either the humoral immunity by activating B cells, and these B cells, some of them become memory B cells, so that in future, if they encounter the same antigen, they're ready to produce antibodies. And then some of them are becoming plasma cells that will produce antibody to that very specific antigen that the T cell recognizes it. This T cell will also mount a cellular immunity where cytotoxic T cells become active, and some of these cytotoxic T cells also turn into memory T cells for potential future attack by this type of antigen. All right, the next concept I would like to talk to you is called clonal selection. And so basically, once a new antigen enters the body, only very few T cells or B cells can recognize that antigen. But these T cells will undergo division in order to produce a clone of cells that can recognize that specific antigen. And so this very first antigen encounter requires to primary immune response, and the expansion of lymphocytes that have recognized it refers to as clonal selection. So expansion of lymphocytes that recognized the antigen. And so therefore there will be long-lived memory cells that will continue to circulate in the body, and if there is future infections with the same antigen, that's the time that these memory cells can now mount a much stronger and faster attack. And now this is referred to as secondary immune response, which is mediated by our immune cells. And so regarding primary immune response and secondary immune response, just think of chickenpox. The first time that we get chickenpox, we are being infected and the body learns how to find it. But in future, anytime you're exposed to it, the body knows how to fight it. So it could take decades, or sometimes even we'll never get the chickenpox again, because memory lymphocytes are now circulating in our blood that know how to destroy the virus in subsequent invasions. Now we keep talking about different antibodies, so next I would like to talk about the functions of different antibodies. So it's the same term to say either antibodies or immunoglobulins, and these are basically immune proteins that help attack different antigens. And there are different classes. The first one is IgM, also known as natural antibodies, because these antibodies are constantly being produced and they are the first antibodies that are secreted during the primary response. So first to secrete during primary immune response, versus IgG, which is the primary antibody that is being produced in the secondary immune response. And so that's one way of knowing, is it the first time that someone has got infected with a specific virus or a specific bacteria, or is it the second time? You check for their IgM and IgG levels. If you see that their IgM level is very high against that bacteria, then that's the first time. If you see their IgG is increased, then you know that this is not the first time. This is the secondary immune response. Now, the other form of antibodies that we have here include IgD, and this is basically found on the surface of B lymphocytes to help recognize antigens. So basically these are receptors on the surface of B cells. Next we have IgA, which is the type of antibody that is found in body fluid secretions. Like for instance, is found in the saliva and in mother's breast milk. And then finally we have IgE, which initiates an allergic reaction. So this IgE is actually receptors that are found on the surface of mast cells. So they have a lot of IgE receptors. And once these IgE becomes active on the surface of mast cells will release histamine, and histamine will mount an allergic reaction. All right, so here I have a structure of antibody shown for you. And as you can see, an antibody has a Y shape. Now there are 4 chains that you can see in this image. 2 short ones, and these are called light chains, and then 2 long ones that are called heavy chains, and heavy chains are being held together by disulfide bonds. Now there are other regions here that I've shown with different colors for you. Some are shown with yellow, and these are actually called variable region because these are the sites that will bind to antigen. And so differences in the variable region makes different antibodies capable of binding to different antigens, versus the constant region, which is shown here in purple color. Constant region is the same in different classes of antibodies. So you know how we have an IgG antibody that binds to, let's say, antigen 1, and we have another IgG antibody that binds to antigen 2. The difference between these 2 IgG antibodies is that their variable region is different, because one is capable of binding to antigen 1 while the other one is capable of binding to antigen 2. But both of these antibodies have the same constant region. Same with other antibodies. If you look at IgA antibodies, for instance, all of them have the same constant region, but it's their variable region that is different, making them specific for binding to different antigens. So now regarding the antigen-antibody recognition, as we just discussed, the variable region of the antibody of both the heavy and light chain confers the ability to bind to a specific antigen. So here, if you pay attention to this image, let me erase all these drawings. So in this image, you can see that the variable region is both on the light chain and the heavy chain. So as we just discussed, the variable region of the antibody can bind to the antigen. Now these variable regions have amino acids that have flexible side chains, as a consequence of which these structures are flexible and can change their shape around the antigen. So therefore, usually one antibody can cross-react to structurally similar antigens and can target closely related pathogens. Regarding the autoimmune disorders, basically autoimmune disorders refer to conditions where our immune system is mistakenly attacking our own cells. And so in other words, the adaptive immunity of our own body is attacking our self-antigen. And so to fight autoimmune disorders, there are different medications that we can give to calm down the immune system. And usually the acute phases are being treated by steroids to tell the body, hey, calm down, everything is all right, you don't need to start attacking the body. Moving on to different immune tissues and cells. So basically we have the primary lymphoid organs and secondary lymphoid organs. So what do we mean by primary and secondary? Primary is basically structures inside which the T cells can develop and mature, but they are not yet exposed to any antigen. An example is bone marrow and thymus. So inside bone marrow, inside the bone marrow, T and B lymphocytes both are formed. B cells stay there until they mature, but T cells move to thymus. And inside there, T cells will mature. They are now ready to start an attack against an antigen, but since they are still not yet exposed in these organs, we refer to these organs as primary lymphoid organs, meaning that the antigen is not yet exposed to the T cells. So one memory aid that I have for you is to remember that bone marrow is the site for B cell maturation, while thymus is the site for T cell maturation. All right, so that was for the primary lymphoid organs. Now these lymphocytes need to at some point get exposed to antigen and mount an immune attack. And those sites that they get exposed to antigens are called secondary lymphoid organs, which includes the lymph nodes, spleen, and tonsils. So let's go over these structures in a little more detail. So thymus is a site where majority of T lymphocytes will mature and start to express either CD4 receptors or CD8 receptors. And we know that cells that have CD4 are called helper T cells, versus cells that have CD8 are called cytotoxic or killer T cells. And CD4 is specific for recognizing MHC2, while CD8 is specific for recognizing MHC1 glycoproteins. Then we have the bone marrow, which is the site for hematopoiesis, where lymphocytes develop from hematopoietic stem cells. B cells will stay in the bone marrow until they mature, and then later on they move to secondary lymphoid organs in order to get exposed to different antigens. T cells, however, after they develop inside the bone marrow, they go to the thymus so that they mature over there. Now moving on to secondary lymphoid organs. So first we have the lymph nodes, and in the circulatory video we've already discussed the function of the lymph nodes, but here let's review it with you one more time. So basically all the collecting lymphatic vessels pass through the lymph nodes, and these structures basically function as filters. So any antigen that is in our circulation will be detected by the white blood cells that are in the lymph nodes, and so consequently these white blood cells can now mount an immune reaction. Spleen, on the other hand, it's made of three different structures. It's made of a red pulp, which is the site of the spleen that is engorged with red blood cells. It's made of a white pulp, which is the site on the spleen that is housing the white blood cells or lymphocytes. And finally, it has the splenic sinusoids, which are lined by macrophages. And as red blood cells are being squeezed through splenic sinusoids, macrophages will recognize those red blood cells that are old or are damaged and will destroy them. Therefore, spleen is the major site for destruction of old red blood cells, old or damaged red blood cells. And then finally we have tonsils, which are basically housing B and T lymphocytes, so B cells and T cells. And these tonsils enlarge in response to certain type of infections, like for instance viral or bacterial infections, mostly at young ages, like for instance in kindergarten children, as a consequence of which if these tonsils enlarge too much, we have to remove them because it could cause obstruction of upper airway. So if you have a friend who tells you my tonsils was removed at an early age, that tells why, because he or she probably had an infection. It got enlarged to a point that the doctors were worried it could cause obstruction of upper airway. Okay, so here next in this table I have provided you with a summary that summarizes all the difference between innate and adaptive immunity. So we just discussed innate immunity is the very first type of immunity that will encounter any entering pathogen or any entering antigen, and these could include physical or chemical barriers, like for instance skin lysozyme or stomach acid. It could include macrophages, it could include neutrophils, eosinophils or basophils, natural killer cells, dendritic cells, complement molecules, interferon for fighting viruses, and C-reactive protein for mounting an inflammatory reaction. This type of immunity is always present with an immediate response within minutes, but it's not really specific nor potent. So this is more like a general response to any type of pathogen or any type of microbe that enters. It doesn't have any memory, so if we get infected by chickenpox today, our innate immunity, if we get infected again 6 months from now, doesn't recognize specifically this antigen, will mount a general immune response. And so therefore there is no clonal expansion. Versus adaptive immunity, which includes the humoral immunity that produces antibodies, or cellular immunity, where there would be cytotoxic T cells that attack any cell that has an abnormal antigen under MHC1. This type of immunity has a very high potency and very high specificity. It has a slower response in compared to innate immunity because it will take some time for it to develop. It does have memory development, so there will be memory T cells and memory B cells that continue in our circulation, and so future infections there would be a faster response. And yes, there would be clonal expansion because T lymphocytes that recognize and the antigen will start to expand in number, T or B lymphocytes. So here I have a table that shows you how hematopoietic stem cells can give rise to different types of blood cells. For this video, I would like to focus more on the immune cells that we are having here. So we just discussed how natural killer cells, these are actually attacking any cancer or viral infected cells by releasing perforin. So these are attacking cancer or viral infected cells. We discussed T lymphocytes are divided into CD8 positive and CD4 positive T cells. CD8 positive T cells are cytotoxic versus CD4 positive T cells are helper T cells that can now activate more CD8 positive, or they could activate B lymphocytes to turn into plasma cells, which are the antibody producing cells. Then there are megakaryocytes that produce platelets. These are required for coagulation. Red blood cells for carrying oxygen and carbon dioxide. Then we have mast cells. We just discussed these mast cells have IgE receptors. So IgE receptors. As a consequence of which, upon their activation, they would release histamine that will induce an allergic reaction. Then we have the granulocytes, including the neutrophils. So we just discussed neutrophils is a component of pus, and that its function is to engulf and kill bacteria by phagocytosis. Basophils, on the other hand, they would initiate an inflammation and allergic reaction by releasing histamine. So their function is very similar to mast cells. So let me write it with a different color. So basophils will release histamine in order to induce an allergic reaction. And then we have eosinophils, which are used for invading parasites. And then finally we have macrophages, which we just discussed. Macrophages are required for phagocytosis or engulfing microbes and other pathogens. Okay, so we are basically done with discussion of this video. There are some questions that I didn't answer during my lecture, so I would like to quickly cover those questions before we go over some practice questions. So first question, how do natural killer cells recognize healthy cells from cancer or viral infected cells? So basically, these natural killer cells also recognize MHC1, similar to CD8 positive T cells. And if these cells notice that the MHC1 on the cell surface is looking abnormal or have something that gives clues that, oh, it's a malignant or virally infected cell, then that's the time that they would release perforin and destroy that cell. What is human leukocyte antigen? Basically, it's the same as MHC protein, except that MHC protein in humans are called human leukocyte antigen. The next question is, describe the process of positive and negative selection in thymus. So we just described that within the thymus, the T lymphocytes become mature, but we didn't describe how these T lymphocytes learn to find antigen and so on. So within the thymus, two processes needs to go on. One, these T cells will have to learn to recognize antigens, and that would be called positive selection. So any T lymphocytes that is able to recognize MHC1 antigens within the thymus, then the thymus will say, okay, you're good enough, you're competent enough to recognize antigens, you will undergo positive selection, meaning that we won't kill you. In the next step, however, all these T lymphocytes that have survived, they would undergo another test. And at this time, thymus will see how much affinity do these T cells have for the MHC1 antigen. Are they crazy about attacking our own antigen? Or no, they're reasonable and they recognize them. If they're crazy about it and attack our self-antigen, then they would undergo negative selection, because the thymus would be like, you're going to attack our own cells, we don't really want you. So they undergo negative selection, meaning that they die. Okay? Now, the problem with autoimmune disorder is that these cells do not undergo negative selection, and so they keep attacking our own cell because they recognize MHC1 and they badly attack it. So that's the reason that people with autoimmune disorders have their immune system attack their own cells. The next question is, what's the function of suppressor T cells? So these are regulatory lymphocytes that dampen the response of B and T cells. And so basically this suppressor T cells are being released following an infection so that it can bring the immune system back to the baseline. It's also used to turn off self-reactive lymphocytes. So if there is a lymphocyte that is very self-reactive and somehow has not gone through negative selection, this suppressor T cell will dampen, will turn it off so that it doesn't attack our own body. So if we have no suppressor T cells, then we are at increased risk of developing autoimmune disorders. The next question is, what is opsonization? Basically, this is the process where a pathogen is being marked with an antibody or complement molecules for destruction. So this is marking of a pathogen or a bacteria for destruction. The next question is, roughly how many different antigen-specific antibodies is produced by each plasma cells? And the answer is, each plasma cell is specific to produce only one type of antibody. All right, so only one type of antibody per plasma cell. We discussed that the variable region is plastic and flexible and can bind different pathogens, but there is still one type of antibody that is being produced by each plasma. Which immune cells are attacked, here's a typing mistake, are attacked by an HIV infection? And the answer is CD4 positive T cells. These are the cells that are being attacked by HIV virus, as a consequence of which now there won't be any more adaptive immunity. Describe the barrier mechanisms of innate immunity in respiratory and GI tracts. So in the respiratory tract, we have cilia and mucous membrane, and the job of these cilia and mucous membrane is to trap the dust particles. Cilia will also wiggle to push up all these trapped particles upward towards the oropharynx so that we can either cough them or swallow them. GI tract, on the other hand, basically this system eliminates most of the ingested pathogens by the stomach acid. So the stomach hydrochloric acid will kill most of the pathogens. But then we also have a lot of benign bacterial flora in the intestinal tract that will prevent growth of the opportunistic bacteria. So the flora in the intestine. And for respiratory system, we had cilia and mucus. What is lymphadenopathy? So basically, once we are having infection, that's the time that our lymph nodes can become enlarged. And you might have noticed that some of the doctors keep checking our lymph nodes behind our ears and under our throat. So this process where there would be enlargement of lymph nodes from an infection is called lymphadenopathy. So this is a palpable enlarged lymph node. The next question is, how are different blood types A, B, AB, and O different? So you know how we hear someone is universal donor and how someone is universal acceptor. So basically, the reason these blood type A or B or AB, these are different It's because of the type of antigen that is present on their cell surface. So blood type A has the A antigen. Blood type B, on the other hand, has the B antigen. Now, anyone that has blood type A will not make any antibody against A, but they would make antibodies against the B antigen. So these patients who have blood type A cannot receive blood type B or AB because their body would attack these blood cells. Same with blood type B. Patients with blood type B, since they have B antigen on their cell surface, they would not make any antibody against this antigen, but they would make antibody against A antigen. So you cannot transfuse someone with blood type B with either blood type A or AB. Now, what happens if someone has blood type AB, meaning that there is both A and B antigen? Consequently, these people will have no antibody that is being formed. And so they are considered as universal recipients because they can receive blood from anyone, A, B, or O. Now, what happens with blood type O? Basically, blood type O, they have no antigen. And so therefore, their body will make antibody against both A and B antigen. So people who have blood type O can only receive blood from other individuals who have blood type O. However, since they have no antigen, they can give blood to anyone. Okay, and this is the reason that blood type O is called universal donor. Now, the other way that MCAT could ask you about these blood types is instead of saying like AA blood, they may say IA. All right, and with blood type O, they show it as II. So here I have shown you different genotypes for different blood type. So blood type O is recessive. And so if someone has blood type A, it could be either AA or AI. Same with blood type B. People who have blood type B, they could be homozygous as BB, or they could be heterozygous, having BO. Patients with blood type AB can have only AB genotype. And then patients with blood type O can only have O genotype, which is II. Okay? What is the composition of pus? We already discussed. It's neutrophils that die from engulfing pathogens, and the remnant of these neutrophils produce a yellowish opaque liquid that is called pus. And then how can neutrophils reach the site of infection from inside the blood vessels? So there are different processes that let a neutrophil exit a blood vessel and migrate to the site of the infection. And here I have listed these processes for you. The first one is called margination, where vasodilation slows down blood flow. Following that, there would be rolling, where the neutrophils binds to the vasculature. Then there would be a tighter binding to the vasculature. Finally, there would be diapedesis, where there would be movement of immune cell through an intact capillary wall. And then finally, the neutrophils will migrate to the site of the infection. All right, now that we are done with these short questions, let's go over some practice question. If you need to take a break, you can go ahead, pause the video. Otherwise, I'm going to read all the questions for you and then come back and answer them. So the first question, antigenic properties of blood types are determined by cell surface carbohydrate. Given this information, which of the following is a true statement? Moving on to the next question. Compared to adaptive immunity, which of the following is true regarding innate immunity? Which of the following properties is consistent with innate immunity? And then the next question is, which of the following cell types is the main activator of adaptive immunity? So all these questions should be easy to answer after listening to this video. And then which of the following cell types produces immunoglobulin? So if you need more time, you can go ahead and pause the video. So for the first question, antigenic properties of blood types are determined by cell surface carbohydrates. Given this information, which of the following is a true statement? Blood type is determined by inherited genes that code for different carbohydrates. Well, if you know genes have DNA, and DNA produces mRNA, and then mRNA produces proteins, which can function as enzymes, structural proteins, channels, or other stuff, right? So blood type is determined by inherited genes. So far that is correct. That code for different carbohydrates? No, our genes don't code for carbohydrates, they code for different proteins and enzymes. Now these proteins and enzymes can alter the structure of carbohydrates, but basically the wording of this choice is incorrect. Choice B, blood type is inherited by enzymes that regulate cell surface carbohydrate binding. That is correct. Cell surface properties of different blood types is inherited maternally. No, it's inherited by both father and mother. We just discussed that the conditions that are being inherited maternally are mitochondrial disorders, but all other genes are being inherited by mother and father. Blood type is independent of genomic coding, and that's incorrect. Our blood type is being inherited from parents to children. Compared to adaptive immunity, which of the following is true regarding innate immunity? So we just discussed how adaptive immunity could be slower, but then it mounts a more specific and a stronger attack, more potent attack. So the answer is compared to adaptive immunity, which of the following is true regarding innate immunity? Well, this one has low potency and low specificity. Versus if we were talking about adaptive immunity, choice C would have been correct. Which of the following properties is consistent with innate immunity? High specificity? No. Active at initial exposure to pathogens? That's correct. They are always ready to mount an immune attack. Develop over a period of days? No, that's more for adaptive immunity. Innate immunity mounts a very rapid attack. And its dependence on clonal expansion of lymphocytes, again, that's a property of adaptive immunity. And then the next question should be very easy. Which of the following cell types is a main activator of adaptive immunity? CD4 positive T cells. We just discussed how after antigen is being picked up, they're being presented to CD4 positive T cells, and now these cells can activate either B lymphocytes or CD8 positive T lymphocytes. And here is the table that shows how adaptive immunity is being activated by the helper T cells. CD4 positive is being the main activator. Then the next question is, which of the following cell types produce immunoglobulins? So CD4 positive T cells activate B cells to turn into plasma cells, and then plasma cells are producing antibody. So the answer here should be plasma cells. Moving on to the last set of questions. Which of the following is the underlying cause of autoimmune disorders? Which of the following tissues do lymphocytes get exposed to foreign antigens? So basically it's telling you which of the following is a secondary lymphoid organ. Which of the following is true regarding the type of immunity that is confirmed to infants upon ingestion of mother's milk? And then the last question is, which of the following antigens is or are present on the surface of red blood cells from universal blood recipients? So back to the first question. Which of the following is the underlying cause of autoimmune disorders? So if you recall, without just looking at the answer choices, autoimmune disorders is due to our immune cells mounting a too strong an attack against MHC1 self-antigens. So reaction of self-antibodies with self-antigens. That would be the correct answer. Which of the following, inside which of these tissues lymphocytes get exposed to foreign antigens? And so we had primary lymphoid organs like thymus and bone marrow, and then we had secondary lymphoid organs like, for instance, tonsils. We had lymph nodes, and then we had the spleen. So spleen would be the correct answer, and these two would not be correct. So choice A only is the site where lymphocytes can get exposed to foreign antigens. And then the final two questions. Which of the following is true regarding the type of immunity that is conferred to infants upon ingestion of mother's milk? So we already discussed that mother milk has IgA. IgD was the type of antibody that is present on the B cells for recognizing different antigens. Now, the next question that you need to answer is this type of immunity active or passive? So passive is when we receive antibodies from a different source than our own body. Like for instance, if we have the horse produce antibody against tetanus, and someone who has tetanus, we inject them with this antibody, it's called a passive immunity. Same goes with infants that drink their mother's milk. The mother's milk contains antibodies that help the infant fight infections. So this is an example of passive immunity from IgA antibody that is present in milk. And then the final question, which of the following antigens is present on the surface of red blood cells from universal blood recipients? So without looking at the table I showed you, recall that universal blood recipients, they would not make antibody against any antigen. And the reason for that is that they have both A and B antigen. So they have both A and B antigen, as a consequence of which they won't attack these antigens. All right, so they have both A and B antigens, and that would be the correct answer. So here, let me review that for you one more time here. So here, the universal blood recipients have both A and B antigen, as a consequence of which, since their body is not making antibodies against either one of those, they can receive blood from anyone. Someone with blood type A, someone with blood type B, or from universal donors. All right, so that concludes our discussion. We have discussed the topics that are listed on the AAMC website regarding the immune system. Hope you found it helpful, and we will see you for the next video.